Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee. Welcome to the Stack Series.
It is wonderful to be back.
Today we are exploring the complex world of stem cell therapies. And honestly, this whole topic feels like walking a tightrope. It really does.
A tightrope. How so?
Well, it's like on one side of the canyon, you have these incredible Nobel Prize-winning scientific breakthroughs. I mean, stuff that sounds like straight-up science fiction. We are talking about rewriting the very code of life. And then on the other side of the canyon, you have strip malls.
Strip malls.
You know exactly what I mean.
Yeah.
I was driving the other day, and I saw one. It was this little clinic, right? And it was sandwiched between a nail salon and a vape shop.
Oh, wow.
And it had this flickering green neon sign in the window that just said stem cells. And then underneath this list, knee pain, hair loss, anti-aging, brain fog.
Right.
It looked less like a state-of-the-art medical facility and more like a place you'd go to buy discount tires.
That imagery it's incredibly stark, but it's also, unfortunately, very, very accurate.
It is.
It creates this massive paradox that you're talking about. We're hearing about medical miracles and snake oil in the exact same breath.
That is the tension. That is what I really want to get into today. Because if you just Google stem cells, you get two completely different realities.
You do.
Reality A is, these high-tech labs curing sickle cell disease for the first time in history. And reality B is a guy in a lab coat in a strip mall offering to inject your spine for $5,000 cash.
That's the perfect way to frame it. It is a paradox. And here we are sitting in February 2026, and the landscape has shifted so dramatically, even just in the last 2 or 3 years.
Keep moving that fast.
It's moving incredibly fast. We are seeing things that were purely theoretical a decade ago, actually saving lives today. But running parallel to that, there is this predatory wellness industry that is it's very, very good at exploiting hope.
Okay, so that's our mission for this exploration today. We need to untangle this mess. We need to separate the biology from the brochure.
A great way to put it.
Because the stakes are just incredibly high. We're not just talking about cosmetic stuff here. We are talking about potential treatments for Parkinson's, for diabetes, for major life-altering conditions.
Exactly. And to understand why the scams work, and more importantly, why the real science is actually so difficult to pull off, we have to start at the very beginning.
At the cellular level.
At the cellular level. We have to look at what we could call the biology of potential.
Right. The master key of the body, I've heard it called that. So when we say stem cell, what are we actually talking about? What is this thing?
Well, in a manner of speaking, we're holding another paradox. A stem cell is a cell with a very specific and very powerful dual superpower.
A dual superpower.
Yes. You have to understand, most of the cells in your body are, well, they're kind of boring.
Okay.
No, I mean, they have a job. A skin cell is a skin cell. A neuron is a neuron. They don't switch careers. They clock in, they do their specific work, and eventually they die.
They're locked in. They've already declared their major, they've graduated, and now they're working the 9 to 5 until retirement.
Precisely. The technical term is that they are terminally differentiated.
Yeah.
But a stem cell is different. Its first superpower is what we call self-renewal.
Self-renewal.
It can make infinite copies of itself. It could just keep dividing and dividing, and it stays a stem cell.
Yeah.
In the right conditions, it's effectively immortal.
Okay, so that's the immortality aspect. It's like a fountain of youth on a cellular level.
In a way. And the second superpower, which is maybe even more important, is differentiation.
Differentiation. So becoming something else.
Exactly. Under the right chemical signals, under the right instructions, it can turn into a specialized cell. It can become a heart cell that beats, a brain cell that fires, or a blood cell that carries oxygen.
I always think of this like, okay, imagine you're playing a game of Scrabble. Most of your cells are the letters already on the board. They're an A or they're a Z. They have a fixed value, a fixed place. You can't just decide to use a Z to spell the word cat. It doesn't work.
Right, it's locked.
But a stem cell, that's the blank tile.
The blank tile. I like that.
It has no value on its own, 0 points, but it's the most valuable tile in the whole bag because of what it could become. It could be an X to get you that triple word score. It could be an E. It completes the word. It has pure potential.
That is a very, very apt analogy. Or, to stick with your university metaphor, the stem cell is the freshman who hasn't declared a major yet.
Right. They're just walking around campus, maybe playing a little too much Frisbee, not sure what they want to do.
But the potential is limitless. They could become an engineer, a poet, a brain surgeon. The potential is there. But, and this is a huge but, not all freshmen have the same potential. And not all stem cells are the same.
Ah.
And this is where the public gets confused and where the scammers really take advantage.
Right, because I think the average person hears stem cell and they just think it's this magic pixie dust that you sprinkle on a problem and it turns into whatever you need it to be. But there's a hierarchy, you said, a pecking order.
There is a strict hierarchy of potency. We absolutely have to distinguish between the real heavy hitters and the specialists.
Okay, so let's start at the top. Who are the heavy hitters?
At the very top of that hierarchy, we have what are called pluripotent stem cells.
Pluripotent.
These are the true blank slates, the true blank tiles from your Scrabble bag.
So these are the ones that can become any of the, 200 plus cell types in the human body?
Any of them. Correct.
Yeah.
If your goal is to make a new liver or retina for the eye or a patch of heart muscle, you need a pluripotent cell. There's no other way.
And historically, this is where the big ethical debate came from, right?
Exactly. Historically, the gold standard for this was the embryonic stem cell, or HESC.
HESC.
These are derived from the blastocyst, which is a very, very early stage embryo, maybe 5 to 7 days old. It's just a tiny ball of cells.
And this is what ignited the firestorm back in the early 2000s.
It was a firestorm. Biologically, these cells are incredibly powerful. They are literally the source code of human life. But ethically and politically, obtaining them requires the destruction of that embryo.
And that led to all sorts of bans, funding restrictions.
And a massive global debate. It really slowed down research in some parts of the world for years. It was this huge gridlock. Science on one side, ethics on the other.
It felt like a dead end for a while.
Yeah.
But then something changed the game completely. You mentioned Nobel Prizes earlier. This is where that comes in, isn't it?
This is the moment.
Yes.
This is the moment everything changed. The discovery of induced pluripotent stem cells. iPSCs. This is what won Shinya Yamanaka the Nobel Prize in 2012. He essentially figured out how to wind back the clock on a cell.
Okay, let's pause on this, because winding back the clock sounds like a nice metaphor, but you mean it literally, don't you?
I mean it quite literally. You have to understand the biological dogma that existed before 2012. Biology had a rule. Time only moves forward. A fertilized egg becomes an embryo, which becomes a fetus, which becomes a baby, and then an adult. Cells differentiate. They go from potential to specific. And we all thought that was a one-way street.
You can't unbake a cake.
You can't turn a hamburger back into a cow. That was the thinking. Once you're a skin cell for life. You can't just decide to be an embryo again.
That's what we thought.
That's what we thought. But Yamanaka, he broke the arrow of time. He took a fully specialized adult cell, just a regular skin cell, a fibroblast from a donor, and he exposed it to 4 specific genetic factors.
Just 4?
Just 4 proteins. They're now famously called the Yamanaka factors.
Okay.
And those factors, they forced the cell to forget its identity. They stripped away all the epigenetic marks. That's like the memory of being a skin cell. And reverted it back to an embryonic-like state. It became pluripotent again.
That is absolutely mind-blowing. It's like taking that guy who's been an accountant for 40 years. You wave a magic wand, and suddenly he's an 18-year-old freshman in college again with his whole life ahead of him.
And he can now choose to become an astronaut or a heart cell. It was a massive, massive breakthrough.
And it sidestepped the whole ethical minefield with embryos.
Completely. And theoretically, you could make them from a patient's own body. So you take a skin cell from a patient, turn it into an iPSC, make new heart cells from it, and put it back in. No immune rejection.
Hold on, though. You make it sound like a software reset, like you press factory restore on an iPhone and it's good as new, but biology is always messier than that, right?
It is.
If I take a skin cell from a 60-year-old, does that new iPSC, does it remember being 60? Or is it truly a newborn cell? Does it carry the baggage of the donor?
That is the billion-dollar question. And the answer is, a little uncomfortably, yes. It carries some epigenetic baggage. It's not a perfectly clean slate. No, that reset process is actually quite violent for the cell. It's a traumatic event. And sometimes mistakes happen. Little errors can creep into the DNA during that reprogramming. Which is why, as we'll get into, using these for actual therapy is not as simple as just injecting the magic juice. There are risks.
Okay, so that's the top tier. Pluripotent cells, the iPSCs and the embryonic cells, they can become anything. But then you mentioned these specialists, the cells that are already in our bodies right now.
Yes, these are the multipotent stem cells, or adult stem cells.
Yeah.
These are the specialists. They live in various tissues in our bodies right now.
Yeah.
The most famous and the most well-established in medicine are hematopoietic stem cells, HSCs.
And hematopoietic just means blood forming.
Exactly. These cells live in your bone marrow, and their only job is to make blood. Red blood cells, white blood cells, platelets. That's all they do.
And these are the cells used in bone marrow transplants, right? Which, that's been a therapy for a long time.
For decades. It is a proven life-saving therapy for things like leukemia and other blood disorders.
Yeah.
If you destroy someone's cancerous bone marrow with chemotherapy, you can transplant healthy HSCs from a donor, and they will rebuild the entire blood and immune system from scratch.
But, and I'm sensing a but here.
But, and listen closely because this is important, an HSC makes blood. It does not make brain tissue. It does not make heart muscle.
It stays in its lane. It's a specialist, like you said.
It stays in its lane. And then we have the other famous type of adult stem cell, mesenchymal stem cells, or MSCs.
MSU.
These are found in fat tissue, bone marrow, umbilical cords. And this, Andrew, this is where the confusion and, frankly, the fraud often takes root.
Why is that? What's so special or so confusing about MSCs?
MSCs are fascinating cells. In the body, their natural job is to differentiate into bone, cartilage, and fat. That's their lane. But in the Petri dish, and especially in the marketing brochures of those strip mall clinics, they are sold as a master key for everything imaginable.
So the clinic claims if I inject an MSC from my fat tissue into my damaged knee, it's going to turn into new cartilage.
That's the claim.
Or if I inject it into a brain with Alzheimer's, it's going to turn into new neurons.
That is the claim. But the actual science, the evidence, suggests something very different is happening.
What's really happening then?
The current thinking is that they act more like managers or mobile drugstores.
Okay. What do you mean by that?
They don't necessarily become the new tissue themselves. Instead, they arrive at the site of injury and they release a whole cocktail of chemical signals. It's called paracrine signaling.
Paracrine signaling.
These signals basically tell the body's existing cells to get to work. They can tamp down inflammation, they can encourage local cells to repair themselves, they can prevent cell death.
Okay, so that's a crucial distinction, a really important one. They aren't the bricklayers building the new wall. They're more like the foreman on the construction site, shouting at the existing crew to work faster and bringing them supplies.
Exactly. Or it's the ambulance arriving at the scene, it's bringing bandages and medicine. And that is therapeutically useful. Reducing inflammation is great. But it is not the same thing as regrowing a brain.
Right.
You cannot just inject a bone marrow cell into someone's spine and expect it to magically know how to become a neuron. Biology is not magic. It's a series of very, very specific instructions. And if the cell doesn't get the right instructions, it won't do the job. Or worse, it will do the wrong job.
This brings us perfectly to what we're calling the science in between. Because, okay, if we have these amazing cells, especially the iPSCs that can become anything, why can't I just go to my doctor today and get a shot to fix my bad shoulder or my failing vision? Why isn't this available at every corner clinic? Why the long wait?
Because moving from the bench, the laboratory, to the bedside is an incredibly difficult, expensive, and frankly dangerous journey. We are facing technical hurdles that are fundamentally different from making regular drugs.
It starts with just making things, right? You called it a manufacturing nightmare.
It is a nightmare. Think about aspirin. Aspirin is a simple chemical. You mix specific ingredients in a giant sterile vat. You press it into a pill. Every single pill is identical.
Right.
You can ship it in a hot truck in the middle of summer. It can sit on a pharmacy shelf for 5 years, and it works the same. Now, try to manufacture a living thing.
It's more like farming than it is manufacturing, I guess.
It's extreme farming. Imagine you're running a factory where the product gets stressed out if the air conditioning hums at the wrong frequency.
Chuckle.
I'm only slightly exaggerating. You have to keep these cells alive.
Yeah.
For one, you have to feed them these incredibly expensive, complex nutrient broths. And you have to make sure every single batch of billions of cells is identical.
And that's the hard part.
It's incredibly hard because cells change. They are living things. They react to their environment.
So if the temperature in the lab shifts by ½ a degree, or the pH of the soup they're floating in is just slightly off, what happens?
The entire batch could be ruined. The cell might just spontaneously decide to differentiate into something you don't want.
Yeah.
You're trying to grow a billion pure neural stem cells. But because the soup was slightly too acidic, half of them start turning into bone cells.
And now the batch is useless.
It's useless. You've lost millions of dollars and months of time.
Yeah.
This is the challenge of GMP good manufacturing practice for cellular therapies. A whole different ballgame.
And that issue of them changing leads directly to the Goldilocks problem you mentioned in our pre-show notes. This part genuinely terrified me when I read about it.
As it should. This is the problem of differentiation. It's a tightrope. If you put a stem cell into a patient, you need it to be differentiated enough so it does the job you want, like becoming a dopamine neuron for Parkinson's.
Okay.
But not so undifferentiated that it's still pluripotent and goes rogue inside the body.
When you say rogue, you mean like cancer?
That's one risk. Or you can get what are called teratomas.
Teratoma.
A teratoma is a type of tumor, but it's a bizarre, horrific one. The word actually comes from the Greek for monster.
Oh, God. That's not a good sign.
It is not. Because that pluripotent stem cell, remember, it has the potential to be anything.
Right, the blank tile.
So if it gets confused inside the body, if it doesn't get the right signals, it might just try to be everything, all at once.
What do you mean everything?
You find these teratomas containing fully formed teeth.
Oh, you're kidding.
No doubt. Fully formed teeth, clumps of hair, bits of bone, muscle tissue, fragments of gut, all jumbled together in this chaotic, monstrous mass.
That is literal body horror. You go in for a cure for your blindness and you end up growing teeth in your eyeball.
It is the absolute nightmare scenario for this field.
Yeah.
And it's not theoretical.
It's happened.
It has. There's a very tragic case that's well documented in the medical literature. It involved a 13-year-old boy in Israel. This was back around 2009, but it remains a critical cautionary tale.
What happened to him?
He had a rare genetic disease, ataxia-telangiectasia. His family was desperate, understandably. They took him to a clinic in Moscow for fetal neural stem cell injections.
So this was stem cell tourism, which we'll talk more about later. They went outside the established regulatory safety net.
They did. They paid for these injections of cells into his brain and into his spinal cord.
And what was the result?
4 years later, he developed these massive growing tumors in his spine. And when the doctors in Israel operated to remove them and then analyzed the tumors, they found something chilling.
What?
The cells in the tumors, their DNA didn't match the boy's. They genetically matched the donor cells from that clinic in Moscow.
So the cure became the cancer.
The cells were not properly controlled. They were not properly differentiated. They just grew unchecked. This is why the FDA and other regulatory bodies are so incredibly strict. People complain about red tape, but that red tape is there to stop you from growing teeth in your brain.
I'm suddenly a big fan of red tape.
You're introducing a living, dividing entity into a human body. It is not like taking a Tylenol. The risks are profound.
And even if they don't form tumors, you have the problem of them just not
Hanging around.
right. They die off. You can't just inject cells into the bloodstream and hope they find their way to a damaged heart like some kind of homing pigeon.
Precisely. This is the delivery challenge. It's a huge hurdle. Cells need a home. They need a structure. In the body, it's called an extracellular matrix.
Okay.
If you just inject a suspension of cells into, say, a stroke cavity in the brain, which is essentially a hole filled with fluid, most of the cells just float away and die within hours. They have nothing to latch onto.
There was a study about this that got a lot of press and then got debunked, right? The rat stroke study.
Yes, this was a few years back. A university put out a press release claiming they had regenerated brain tissue in rats after a stroke using stem cells. The headlines were amazing. Hole in brain filled with new tissue.
I can see why that would get attention.
But when other scientists looked closely at the actual data, not the press release, they saw that the researchers had used a scaffold. They mixed the cells with tiny biodegradable particles to hold them in place.
So what was it that actually filled the hole in the brain?
The new tissue that filled the hole was mostly just the scaffold.
So it wasn't the cells rebuilding the brain, it was more like they were just filling a crack in the drywall with spackle.
A very good analogy. The structural fill was there, but the actual neural connections, the function, wasn't really restored in the way the headlines implied.
So they just plugged the hole.
They plugged the hole. And this is why we're seeing so much brilliant work now on things like hydrogels and 3D printed scaffolds. It's like building a biocompatible spider web for the cells to live on inside the body so they can survive long enough to actually integrate and connect with the host tissue. We're essentially trying to build synthetic microorgans to hold the cells.
Okay, so just to recap the challenges. The manufacturing is an industrial nightmare. The cells can turn into monster tumors if you're not careful, and you need to build them a house to live in or they just die. It sounds almost impossible, and yet We are seeing approvals. We are seeing real wins.
We are, and this is the truly exciting part of where we are right now in 2026. The last 3 years have been monumental for the field. The science has finally matured enough to clear some of these hurdles.
So let's talk about those validated wins. What is actually real? If I walk into a major hospital today, not a strip mall, what can I actually get?
Before we list them, we need to clarify what approved really means, because the scammers love to use official-sounding language to confuse people.
Okay.
There's a massive difference between an IND and a BLA.
Okay, let's unpack this acronym soup for everyone. What's an IND?
IND stands for Investigational New Drug. This is just a permission slip from the FDA. It means they've reviewed your preliminary data and have given you permission to test your product in humans.
So it's permission to start a trial.
That's it. It is not an approval. It does not mean the drug works or is even safe. It just means you're allowed to start finding that out.
But I bet clinics will put FDA authorized or FDA registered on their website when all they have is an IND, right?
All the time. It is a classic marketing trick. We are operating under an FDA protocol. It sounds official, but it just means they filed the paperwork to start testing. The real approval is a BLA.
And BLA is?
A biologics license application. This is the diploma. This means the testing is done, the data has proven the therapy is safe and effective for a specific condition, and you now have permission from the FDA to market and sell it as a medicine.
So who has the BLA? Who has actually graduated?
Well, the biggest headline of the decade, without a doubt, has to be for sickle cell disease.
Casgevy and Lyfgenia.
Yes. Approved in late 2023, but really starting to roll out to patients in 2024 and 2025. This is the definition of science fiction becoming reality.
So just remind us, what is sickle cell?
Sickle cell is a brutal genetic disease. It's caused by a single typo in your DNA, one wrong letter in the gene for hemoglobin.
And that one typo causes all the problems.
It causes your red blood cells, which should be round and flexible, to become hard and shaped like sickles or crescents. They get stuck in small blood vessels, causing these episodes of immense, agonizing pain, organ damage, and a shortened lifespan.
And Casgevy uses CRISPR, the gene editing tool, to fix this.
That's right. But let's walk through what the patient actually has to go through. It is not just a simple shot. It's a long and difficult process.
Okay.
First, they take the patient's own blood stem cells, those iPSCs we talked about, out of their body. They harvest them from their blood.
So it starts with their own cells.
Yes. Then they send those cells to a highly specialized lab. In that lab, they use the CRISPR gene editing technology to physically go into the DNA inside those cells and edit it.
They're literally rewriting the patient's genetic code.
They are. In this case, they're not fixing the original typo. They're doing something even cleverer. They're breaking the gene that turns off fetal hemoglobin.
Fetal hemoglobin.
You see, when we are babies in the womb, we have a special, super efficient kind of hemoglobin that grabs oxygen really well. After we're born, a genetic switch flips, and our bodies start making adult hemoglobin.
And for sickle cell patients, that adult hemoglobin is the broken version?
Exactly. So Casgevy goes in and it breaks that off switch. So the edited cells, when they're put back in the body, they revert to making the healthy fetal version of hemoglobin again for the rest of the patient's life.
That is just an incredibly elegant solution, but you said the process is hard on the patient.
It is brutal. While their cells are off being edited in the lab, which can take weeks, the patient has to undergo what's called myeloablation.
And that is?
That means high-dose chemotherapy to completely kill off all the remaining defective stem cells in their bone marrow. It completely wipes out their immune system.
So you have to destroy the old, broken factory before you can install the new one?
Yes. And during that time, the patient has to live in strict isolation in a hospital. If they catch a common cold, they could die. It's incredibly risky. And then, once their marrow is empty, they infuse the newly edited supercells back into the patient's bloodstream. The cells naturally find their way back to the bone marrow, set up shop, and start producing healthy, round, red blood cells. It's a functional cure.
A functional cure. It's grueling, it costs millions of dollars, but it works. That is the peak of legitimate stem cell and gene therapy.
It is the absolute pinnacle of where we are today.
Okay, what else is on the approved list? There was one for cancer patients, right? Omasurge.
Yes, Omasurge was approved in April 2023. And this addresses a very specific problem with cord blood transplants.
Okay.
Umbilical cord blood is a great source of stem cells for cancer patients who need a transplant because the immune matching doesn't have to be as perfect as with bone marrow. But there's a catch.
What's the catch?
The cells from cord blood are a bit sleepy. They're immature. When you transplant them, it can take a long time for them to boot up and start making new white blood cells.
And in that gap, while they're booting up, the patient has no immune system.
Exactly. They are incredibly vulnerable to deadly infections. It's a very dangerous period.
So what does Omasurge do?
Omasurge is a technology that basically wakes the cells up and multiplies them in the lab before they're infused into the patient. It's an expansion technology. So when the cells go in, they're ready to work immediately. The recovery is much faster.
It shortens that window of vulnerability.
It saves lives by closing that window. It's a huge step forward in transplant medicine.
And then there was the big one from December 2024, Ryoncil. Why was that one so significant?
Ryoncil, or remestemcel. This one is so important because it is the first FDA approval for a product made of mesenchymal stem cells, MSCs, for this specific use.
The manager cells we talked about.
The manager cells. And it treats a terrible condition called pediatric graft-versus-host disease, or GVHD.
We need to explain GVHD because it sounds terrifying.
It is. Imagine a child gets a bone marrow transplant to cure their leukemia. The transplant works, the cancer is gone. But the new immune system, which came from the donor cells, looks at the child's own body and says, "You are foreign. You are the enemy."
So it's a reverse of an organ rejection. The body isn't rejecting the organ is rejecting the body.
Precisely. The new immune system starts attacking the child's skin, their gut, their liver. The child gets horrific rashes, severe diarrhea, liver failure. It's devastating.
And this happens after they've already been through chemo and a transplant?
Yes. High-dose steroids can usually stop it, but in cases where steroids fail, the mortality rate is tragically high.
And Ryoncil stops that attack.
It uses MSCs, remember, the managers that calm things down, to modulate the immune system. It basically tells those angry, attacking T cells from the donor to stand down. It dials down the inflammation. The fact that this got approved is a huge validation for MSCs, when they are used correctly, for a very specific target with rigorous data to back it up.
Okay, so we have a functional cure for sickle cell, a way to make cancer recovery safer, and a treatment for this horrible transplant complication in kids. These are real FDA-approved, insurance-covered treatments.
Yes, they are the real deal.
But that's not what I see on the billboards on the highway.
No, it is not.
I see ads for stem cell facelifts, or regenerate your knees without surgery, or, and this is the saddest one to me, treatments for autism or dementia or Lou Gehrig's disease. So let's get into the Wild West.
Wild West is a good term for it.
Because if the real science is this hard chemotherapy, isolation, million-dollar labs, how is a clinic next to a vape shop possibly doing it?
The simple answer is they aren't. This is the wellness scam ecosystem, and it is vast. We are talking about thousands of clinics operating in what used to be a regulatory gray zone, or in many cases, just blatantly breaking the law.
How do they get away with it? If the FDA is so strict that they're worried about teeth growing in spines, how is there a clinic down the street offering stem cells for erectile dysfunction?
For a long time, they used a very clever legal loophole. An argument called the same surgical procedure exception.
The same surgical procedure exception. What does that mean?
Okay, so the clinics argued this. We are taking the patient's own fat tissue via liposuction. We're spinning it in a centrifuge to isolate the cells, what they call the stromal vascular fraction, or SDF, and then we are injecting those cells back into the same patient on the same day. Therefore, they argued, we aren't creating a drug. We are just performing a surgical procedure, and the FDA regulates drugs, not the practice of surgery.
I can see the logic there. From a certain point of view, it's my body, my cells. I'm just moving them from point A to point B. If a surgeon takes a skin graft from my leg and moves it to my arm, the FDA doesn't get involved in that.
That was their exact argument. But the courts have finally and decisively disagreed, and this is a major update for our listeners. In September 2024, the Ninth Circuit Court of Appeals ruled in a landmark case, United States v. California Stem Cell Treatment Center.
So play the judge for me. What did the court say?
They said, essentially, no, that argument doesn't fly. When you process that fat, when you spin it down and isolate the cells, you are altering them significantly from their original state. You are manufacturing something.
So it's not just moving tissue anymore.
And more importantly, the court said, when you then claim that these processed cells can cure a disease, when you market them and say, this injection of fat cells will cure your Parkinson's, you have created a drug. You are making a medical claim. Therefore, it is a drug, and it needs to go through the full FDA regulation and approval process like any other drug.
So the loophole is officially closed?
Legally, yes. That ruling was a stake in the heart of that argument. It has empowered the FDA and the Federal Trade Commission, the FTC, to crack down much harder on these clinics.
And we saw that with the Stem Cell Institute of America case in 2025, right?
Yep. Correct. The FTC went after them hard and fined them $5.1 million for deceptive marketing. They were basically a franchise operation, training a whole network of chiropractors and clinics to sell these unproven treatments for joint pain, making all sorts of false claims. They were selling false hope at a premium price.
Okay, so for the listener who might be tempted, because look, chronic pain is real, desperation is real, what are the specific red flags? How do I know if I'm walking into a legitimate research center or a scam?
There are 4 major red flags, 4 signs that you should turn around and walk out the door.
All right, let's list them. Number 1.
Pay to play. This is the biggest one. If you are asked to pay thousands of dollars for the treatment itself, especially if they call it a clinical trial or a study, run.
Wait, really? I thought clinical trials cost money to run.
They do, but they are funded by biotech companies or by the government, like the NIH. They are paying to get your data to see if their drug works. They do not charge the patient for the experimental drug.
So if anything, they should be paying me.
Legitimate trials often pay for your travel, your lodging, your time. They want your participation. If a clinic asks you for $10,000, $15,000, $20,000 to join our patient-funded study, it is almost certainly a scam.
They're just using the word study to make it sound scientific.
Exactly. They're making you feel like you're accessing cutting-edge science, but really you're just a customer buying an unproven, unregulated product. Real science does not ask for your credit card.
That's a huge one. Okay, red flag number 2.
Testimonials over data. Go to their website. Is it filled with glossy, highly produced videos from Brenda in Ohio, who says her knees feel 20 years younger?
Right.
But are there any links to actual peer-reviewed studies in reputable medical journals? Is there any hard data, any charts, any control groups?
But Brenda looks so happy in the video. Why shouldn't I believe her?
Because the placebo effect is incredibly powerful, especially with subjective things like pain. If you pay $10,000 for a treatment, your brain desperately wants it to work. You release natural endorphins. You might feel better for a few weeks, but that is not tissue regeneration. Anecdotes are not evidence.
Brenda might feel better because she just spent 10 grand and doesn't want to admit to herself that it was for nothing.
That too. Real science has boring statistics and error bars. Scams have emotional stories. Look for the data.
Okay, number 3.
The cure-all claim. Biology is specific. A cell that is programmed to fix a heart does not also fix a brain. If a clinic tells you that the exact same vial of cells from umbilical cords can be used to treat your arthritis, your aunt's Alzheimer's, your nephew's autism, and also give you a youthful glow, that it's biologically impossible.
It's the magic elixir pitch from the 1800s, just wrapped in a lab coat with sciencey words.
It is pure snake oil. Different diseases require different cells and different delivery methods. There is no one-size-fits-all magic bullet.
And the last one, number 4.
Celebrity endorsements. We have to be really wary of this in our culture. Timothy Caulfield, a great researcher in this space, calls it science-ploitation.
Science-ploitation.
Just because a famous athlete, a quarterback, or a podcaster with millions of followers says they got stem cells in Panama and they feel younger than ever, that does not mean it works. They are often paid for that endorsement, or they are just as susceptible to the placebo effect as anyone else. Science is not determined by Instagram followers.
And the consequences of falling for these scams. It isn't just losing money, is it? It's not just, oh, well, I wasted 5 or 10 grand. People get seriously hurt.
People get blinded. There have been several documented tragic cases of patients going blind after getting stem cell injections into their eyes for macular degeneration. The cells were unproven, they were injected improperly, and they basically turned into scar tissue on the retina, detaching it.
That's horrifying.
People get severe infections, like spinal abscesses, because these backroom labs and strip malls are not held to the same sterile standards as a hospital operating room. And most insidiously, people delay getting real, proven medical care.
That is the real tragedy.
It is. If you're trying to treat your cancer with regenerative wellness shots from a clinic instead of with chemotherapy or surgery from an oncologist, you might miss the window where your cancer was actually treatable. You are buying false hope at the potential cost of your life.
That is heavy. It really highlights why all that red tape is there. It's not to be mean, it's to keep us safe.
It's to keep us alive, exactly.
Okay, so we've covered the legitimate, approved wins. And we've covered the dangerous scams. But what about the stuff that is almost there, the real frontier? Because I know there's a tremendous amount of excitement right now, specifically around Parkinson's disease.
This is where my optimism comes roaring back. The last couple of years, 2025 and 2026, have been incredible for Parkinson's research. We are finally, after 20 years of hard work, seeing the fruit of that labor in clinical trials.
Parkinson's seems like almost the perfect target for a stem cell therapy, doesn't it? Because it's not a mystery. We know what the problem is.
It's a very specific problem. It's an engineering problem, in a way. Parkinson's is largely caused by the death of one specific type of cell, dopamine-producing neurons in a part of the brain called the substantia nigra.
The dopamine factories shut down.
They shut down. So if we can use stem cells to build new healthy dopamine factories and plant them in the right location, theoretically we can restore that function and treat the symptoms. We don't need to rebuild the whole brain, just that one critical circuit.
So tell us about the two major trials that everyone is talking about. It almost feels like this friendly race between two different technologies.
It is a friendly competition, and both have published major results recently in the journal Nature, which is a huge deal. First, we have the Kyoto Trial, led by the pioneer Dr. Jun Takahashi in Japan.
And this trial used the iPSCs, the Nobel Prize-winning method of winding back the clock.
Yes. They used iPSCs derived from the blood of healthy donors. They carefully programmed those cells in the lab to become dopamine precursors, essentially baby dopamine neurons. And then they surgically implanted them into the brains of 7 patients with Parkinson's.
Where exactly in the brain do they put them?
They're placed very precisely into a region called the putamen, which is a key part of the brain's movement control center. It's the area that's starved for dopamine.
And what did they find? What were the results?
First, and most importantly, it was safe over 2 years. No tumors. That was the big fear with iPSCs, remember, the teratoma risk. But their process was clean.
That's a huge relief.
It is. And on PT scans, which can measure dopamine activity in the brain, they saw a 44.7% increase in dopamine activity in the transplanted areas.
Almost a 50% boost. That's huge. What did that mean for the patients?
It meant the cells survived, they integrated into the brain, and they started doing their job. They were making dopamine. And clinically, many of the patients showed meaningful improvements in their motor symptoms. They could move more freely with less medication.
That's incredible. Okay, and what about the second big trial?
That is the Expedite-2 trial, which was led by Dr. Claire Henchcliffe and conducted in the U.S. and Canada. They used the older method. They used embryonic stem cells, HESCs.
The ones from an embryo, specifically a cell line that was created way back in 1998, right?
Right. It's a very well-characterized vintage cell line, you could say. Scientists know exactly how it behaves, which gives them a lot of confidence in its safety.
And what were their results?
They treated 12 patients. The group that received a higher dose of cells saw a really significant improvement, an average of 23 points on the standardized Parkinson's disease rating scale. For patients, that's a very meaningful difference in their daily quality of life. Less tremor, less stiffness.
But, and I know there's always a but, with you, what's the catch? Is this a cure for Parkinson's?
We have to be so careful with the word cure. The important nuance here is that these cells replace dopamine. And that is fantastic for the motor symptoms of disease, the movement, the shaking, the walking. But Parkinson's is a much more complex systemic disease than we used to think. It also causes many non-motor symptoms, things like depression, sleep disorders, cognitive issues, brain fog. And those problems don't necessarily respond just to dopamine replacement.
So it can fix the tremors, but it might not fix the brain fog or the sleep issues?
That's the current thinking. So these transplants are likely to be a supercharged biological version of the standard drug, levodopa. It's a continuous, steady supply of dopamine made by your own new brain cells, rather than taking pills every few hours that cause peaks and valleys.
So it's a massive upgrade in treatment, but not a total cure for the underlying disease process that's still going on.
Exactly. But for a patient who can't hold a cup of coffee without spilling it, or can't button their own shirt, getting that motor control back, that's life-changing.
Absolutely. It's a massive, massive step forward. Are there other frontiers, other diseases where we're seeing this kind of progress?
Definitely. The other one that's just as exciting is diabetes type 1.
Another missing cell disease.
Precisely. In type 1 diabetes, the body's own immune system attacks and destroys the islet cells in the pancreas that are responsible for making insulin. A company called Vertex Pharmaceuticals has a trial, VX-880, that is just stunning.
What are they doing?
They are manufacturing functional islet cells from pluripotent stem cells in the lab and then infusing them into patients.
And the results?
In the latest data update, 83% of the patients in the trial achieved insulin independence.
Wait, just pause on that. They don't need to take insulin shots anymore?
Correct. Their bodies are making their own insulin again in response to meals.
Yeah.
The factory has been rebuilt. They're essentially cured of the need for daily insulin injections. Now, they do still need to take immunosuppressant drugs so their body doesn't attack the new cells, which is a significant trade-off. But for many, it's a worthwhile one.
That is, wow, that is the dream for every type 1 diabetic. That changes everything.
It is a game changer. And one more, a fascinating one, in the world of IVF and fertility. A company called Gamito, working with another company called reprogrammed, just got FDA permission to start phase 3 trials in February 2025. This is a big deal.
What's the technology? What are they doing?
They are using iPSCs to create ovarian support cells. They then co-culture these lab-grown support cells with immature eggs taken from a woman going through IVF. The idea is that these support cells help the eggs mature outside the body.
So it could make the IVF process easier on the woman.
That's the hope. It could make IVF cheaper, faster, and require fewer of those powerful hormone injections that women have to take to mature their eggs. It's a product called Fertillo.
So we're using stem cells to help create life at the very, very beginning.
Exactly. And it's significant because it's the first therapy made from iPSCs to hit phase 3 trials in the United States. It's a real milestone for the whole field.
It really feels like we're at a tipping point, but as these therapies become real, the ethical questions and the patient access questions get stickier, don't they?
They do. We talked about the outright scams, but there's also the very confusing issue of right to try.
Yeah, I've heard this term a lot. It's the idea that, if I'm dying from a terminal illness, I should have the right to try anything, right? Even if the FDA hasn't stamped it with approval. It's my life. Let me roll the dice.
That is the powerful, emotional core of the argument. And in the United States, right to try, laws do exist at both the federal and state level. But there is a massive public misunderstanding of what they actually do.
People think it means they can try anything they want.
People think right to try means they have the right to go to a strip mall clinic and try some magic water that isn't regulated. But it doesn't mean that at all.
What does it actually mean?
Legally, right to try only applies to drugs that are already in the official FDA pipeline. The drug must have already passed phase 1 safety testing. It allows a terminally ill patient to potentially access an experimental drug, but not an imaginary or unregulated one. It does not give you the right to be scammed.
Ah, so it's not a free-for-all. The product still has to have some level of scientific vetting.
A significant level. And this leads to a debate among ethicists about weak paternalism. On the one hand, we want to respect a patient's autonomy, their right to choose. But on the other hand, if a patient is desperate and a clinic is actively lying to them about the science and the data, is that a truly free choice? Or is it just exploitation?
It feels like exploitation. You can't give informed consent if you're not being given the correct information.
Most experts and regulators would agree with you. And this is why we're starting to see regulation tighten, not just in the U.S., but globally.
The stem cell tourism issue. We mentioned the Israel-Moscow case earlier, but for years, people have been flocking to clinics in Mexico, Panama, China.
They have, but the walls are closing in. China introduced strict new regulations in May of 2026, just a few months ago from our perspective, cracking down on how hospitals and clinics can use these technologies. They realized that a few bad actors were damaging the country's entire scientific reputation.
You're cleaning house.
They are. And Mexico and Australia have also significantly tightened their rules. It's becoming harder and harder for the scammers to find a place to hide. The days of the Wild East or the Wild South where anything goes are slowly coming to an end.
That's very good news. So let's bring it all home for someone listening right now. If I'm listening to this and I have a condition or my parent has a condition and I'm desperate and I'm Googling stem cell therapy, what is my practical checklist? How do I stay safe?
Okay, here is your survival guide.
Mm-hmm.
4 questions you must ask. Write this down. First, check the status. Is the therapy FDA approved? Ask for the specific name. If it's not, ask if it's being done under an FDA-authorized clinical trial. If they say yes, ask to see the IND number.
The investigational new drug number.
Yes. If they can't provide one, that's a huge problem. Second, verify the trial. Don't just take their word for it. Go to the official U.S. government website, clinicaltrials.gov. Every legitimate trial in the country is registered there. Search for the trial number they gave you or the clinic's name. If it's not on that website, it's not a real trial, period.
Okay, that's a concrete action anyone can take.
Third, follow the money. We said it before, but it's the most important. Are they asking you to pay for the treatment? If the answer is yes, it's almost certainly a scam. Real research is funded. They pay you, or at the very least, it costs you nothing. And the final check. Get a second opinion. And this is critical. Get that second opinion from a qualified doctor or specialist who is not affiliated with the clinic selling you the treatment. If the clinic ever says something like, don't tell your regular doctor, they just won't understand this cutting-edge science, that is a giant flashing neon red flag.
Don't tell your doctor is basically the don't tell your parents of the medical world. It's never a good sign.
Never.
Yeah.
Real science invites and loves scrutiny. Scams hate it and hide from it.
Okay, so let's just try to unpack this one last time. We started this whole journey with the paradox. The miracle on one side, the snake oil on the other. It really seems like the takeaway is that the miracles are coming, but they are slow, they are specific, and they are painstakingly, boringly regulated. And the snake oil is fast, it claims to be broad, and it sounds very exciting.
That is the perfect summary. Real science is hard work. It's slow. It involves building scaffolds and meeting GMP manufacturing standards and dealing with years of failure in the lab. But when it finally works, like it is now for sickle cell, like it's starting to for Parkinson's, it changes human history. It changes what it means to be sick.
It really does. It's this whole journey from the biological magic of a single cell that can become anything, to the years of hard work by scientists like Takahashi and Yamanaka, to the caution and skepticism we all need to have against those who are just exploiting hope.
And that caution, that skepticism, is what protects the future of the entire field.
Yeah.
We need to clear out the weeds so these incredible flowers can actually have the space to grow.
Well said. Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content and support us on Patreon.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.